Greetings for the day. Myself, Dr. Akshay Chandra Mukhe, currently working as Research Associate at Vinayaka Missions Shankaracharya Dental College, Tamil Nadu, and PhD Scholar at Singagar Dental College and Hospital, Pune. The topic for my today's presentation is Epigenetics in Oral Neoplasia, Unravelling the Cause and Effect Conundrum. Contents for today's presentation is Introduction, Genetics and Epigenetics, Basics of Epigenetics with examples in Oral Neoplasia, Techniques to study epigenetics, and conclusion. At the end of this presentation, we are expected to know what is epigenetics, types of epigenetic mechanism, and how epigenetics is related to oral neoplasia. As we all know that the neoplastic process or carcinogenesis is driven by somatic mutations or somatic changes that can be attributed to viral introduction of the oncogene, copy number changes, rearrangements. And there is a hidden factor which governs the gene expression and silencing, which is epigenetics. And therefore, we must know the exact mechanism or how epigenetics work. Carcinogenesis attributed to genetic alterations and epigenetic alterations. Genetic alterations can be point mutations, single nucleotide polymorphism, copy number variations. Again, there can be three types of copy number variations, that is loss of heterozygosity, homodeletion, and gain copy number. Whereas in epigenetic alteration, there is three basic mechanisms: DNA methylation, microRNA, and histone modifications. So Conrad Weddington is the one who has coined the term epigenetics. And the simplest definition that I find for epigenetics is given by Wu and Morris, that is the study of changes in gene function that are mitotically and or meiotically heritable and that do not entail a change in DNA sequence. So we must remember that there is no change in the DNA sequence or the DNA base pair in the epigenetic mechanism, which is dynamic and reversible. So the epigenetic mechanism is dynamic and reversible. So when we see a human cell, it contains 46 chromosomes, that is 44 autosomes and 2 sex chromosomes. These all cells have similar DNA, but they look differently, they function differently. So what is the reason behind it? So when let's take an example of ameloblast and odontoblast. So now ameloblast and odontoblast have similar DNA content, but they function differently, they look differently. So according to the similar genetic component or the DNA content, both cells should secrete enamel and dentine, but this doesn't happen. So during the developmental process or during the development of ameloblast, what we see is that there is some epigenetic modifications due to which there is silencing of the genes required to function as odontoblast and expression of the genes committed for ameloblastic function and traits. On contrary, when we see development of odontoblast, there is some epigenetic modifications, there is silencing of the genes required to function as ameloblast, and there is expression of the genes committed for odontoblastic function and traits. So these are the reason why an ameloblast will function like has a function to secrete as enamel, and odontoblast will only secrete dentine. Basics of genetics in brief. So the double-stranded DNA is coiled around the histone protein, and these histone proteins are packed together to form the chromosome. There are several chromatin domains. There is heterochromatin and euchromatin. The basic difference between them is heterochromatin is transcriptionally inactive, whereas euchromatin is transcriptionally active. And heterochromatin can be of two types, that is constitutive and facultative. So constitutive ones are the compulsive heterochromatins that they will be never accessible for transcription process. Whereas when we talk about facultative one, on certain instances, it can function as euchromatin. So there is the central dogma. We see that DNA is transcribed to RNA, and the reverse is also possible. That is called as reverse transcription. And then this RNA can be translated to protein. So let us understand what is the epigenetic mechanism behind in oral neoplasia. So what we know is the central tendency, that is the genetic contribution, is in neoplasia. By mutations of oncogene and tumor suppressor gene, there can be gain or loss of functions of gene and altered expression of gene, which is the central cause. Then there's when we see genetics, there is regulation of genome activity, series of genetic factors that can be cascade of critical transcription factors and regulation of gene expression to promote the programming of transcriptional events. This all leads to development of the tissue and organism. But when there is an alteration or abnormality in this process, there will be formation of pathology or abnormal development. Now, evolving concept says that the epigenetic mechanism will have an influence on the genetic expression, that is expression or the silencing, which is responsible for tumor initiation and progression. Now, this is a very classical example of Knutson's two-hit hypothesis studied in retinoblastoma gene. The first example, we see that there is an hereditary where there is paternal and maternal chromosome. The first mutation is already in the germline mutation, and the second mutation is the later in the life, which can be an epigenetic event due to which there is formation of cancer. When we see the second example, there is three possible mechanisms for development of the cancer. First, there can be a true genetic mutation of one of the chromosome. Second hit can be due to the epigenetic event. Therefore, there will be a cancer progression or cancer initiation. In the second example, we see that the first hit is due to the epigenetic mutation or the epigenetic alteration, and the second hit is again due to the epigenetic mechanism, and therefore there is development of the cancer. The third example or the third mechanism can be the first one is the epigenetic mutation or the epigenetic alteration, and the second one is the true genetic mutation, due to which there is development of cancer. So this is a crucial or an example where we can understand how genetic and epigenetic can play into for the development of cancer. So epigenetic changes can be responsible for areas of pathology like squamous cell carcinoma, odontogenic tumor, salivary gland tumor, sarcomas of oral cavity. Lifestyle factors and other factors can also modify the epigenetic patterns like diet, obesity, physical activity and stress, tobacco, alcohol infections, environmental pollutions, and psychological stress. So describing or giving examples for each and every parameters will be out of the scope of this presentation. So I'm just taking the diet factor to give some example. So here we see that there is epicatechin, which is present in the green tea. It acts as an anti-carcinogenic effect because of the DNA methylase transferase inhibitor and histone acetyl transferase inhibitor, and it also modulates the microRNA. So these are the epigenetic event which makes it an anti-carcinogenic in nature. When we talk about curcumin, there is DNA methyltransferase inhibitor and microRNA modulator, due to which it is again an anti-carcinogenic one. So when we talk about alcohol or alcoholic beverages, it increases promoter hypermethylation, which poses an increased risk for oral cancer or cancer. Now let us study each mechanism of epigenetics, that is DNA methylation, histone modification, and microRNA in detail. So when we talk about DNA methylation is a biological process by which methyl groups are added to DNA molecule. Methylation can change the activity of DNA segment without changing the sequence. So this is an important point that one must remember. When located in a gene promoter region, DNA methylation typically acts as repressor of gene transcription. So there are these key processes which is governed by DNA methylation, genomic imprinting that we see in X chromosome inactivation, which has been given by the Lyon's hypothesis, and that has been seen in as a bar body in the females. Next is carcinogenesis and aging. Now let us talk about the promoter region. When we talk about the promoter region, it's a DNA upstream of gene where relevant proteins such as RNA polymerase and transcription factors can bind to initiate transcription of the gene. And the resulting transcription produces an RNA molecule such as messenger RNA. So here there is an upstream strand and the purple color RNA polymerase. When this purple color RNA polymerase attaches to this strand, upstream strand, there will be the transcription process starts. And when there is a stop codon, the transcription process will end. So the resultant of there will be production of a messenger RNA. Now DNA methylation will always take place in CPG sites or CPG islands of the DNA region or the DNA strand. So what are these CPG islands? These are nothing but the cytosine and guanine molecules of the DNA, and they are always like these CPG islands are always read from 5' to 3' end. So methylation or the methyl group has been added to cytosine molecule, but in the CPG islands which reads from 5' to 3' end. It is always in the 5' to 3' end. It is never in the reverse direction. From 3' to 5', it's never in this reverse direction. So this we must remember. So here we see that there is this DNA base cytosine where it has been catalyzed by various enzymes, that is DNA methyltransferases, DNA methyltransferase 3A and 3B. So when this cytosine, this H is being replaced, this hydroxyl group has been, hydrogen group has been replaced by the methyl group, and it forms 5-methyl cytosine. So this is the methylation process. So one must remember that whenever there is methylation, there is tightening of the DNA molecule or packing of the DNA molecule around the histone protein. And when there is tightening or packing of this histone DNA strand around the histone protein, there will be silencing or a silencing of the gene because it is not available for transcription. On contrary, when we see that there is unmethylation, there will be unpacking of this DNA around the histone protein. So therefore, this DNA strand will be available for transcription process. And then there will be expression of gene. So here there are certain instances where we can see that the oncogene, which is responsible for carcinogenesis, and tumor suppressor gene, which will suppress the carcinogenic process. So here we see when there is hypomethylation of oncogene, there will be the gene will be expressed, then there will be carcinogenesis. When the oncogene is hypermethylated, there will be repression or silencing, due to which there will be anti-cancer effect. When tumor suppressor gene is hypomethylated, there will be expression, and then there will, because of that, there will be anti-cancer effect. And tumor suppressor gene, when they're hypermethylated, there will be silencing, due to which there will be result will be carcinogenesis. When we see oral cancer, that is, the significant differences is seen between the epigenetic signatures, where the tumor sample exhibit increased genome-wide hypomethylation and also hypermethylation of the promoter region. So due to which, when we see hypomethylation, there will be increased chromosome instability. And when we see hypermethylation in the tumor suppressor gene, when they're methylated, it impedes the access of transcription factors to the segment and implicit the expression of specific gene expression. So both mechanisms are studied in carcinogenesis, that is hypermethylation and hypomethylation. Now, when we talk about the tobacco and alcohol consumption, so these are the risk factors, like such as tobacco and alcohol consumption, and also chronic inflammation of the oral mucosa have been linked to dysregulations in the epigenetic pattern regarding the methylation status. So smokers have been associated with an increased global hypomethylation. So a person who is having a increased global hypomethylation is more prone for carcinogenic event. Now, these are the examples which I have taken from different literature, where we see there is hypomethylation of chloride intracellular channel protein 3, which acts as an oncogene, which is studied in mucoepidermoid carcinoma. Hypomethylation of cervivin, which acts as apoptosis marker. It is studied in oral squamous cell carcinoma. Likewise, hypermethylation of P16E-cadherin is studied in chondrosarcoma. And the hypermethylation of P21 as tumor suppressor gene is studied in amyloblastoma and adenomatoid odontogenic tumor. To name, these are the examples where we have studied hypermethylation and hypomethylation. There are other examples as well, which you can find in the literature. So the next is histone modifications. Now, coming to histone modifications, histones are the proteins. On this protein, there is binding of the DNA, and these histones are always in the octamers. And the histone protein and the DNA molecule is called as a nucleosome bead. So histones are small protein with a globular domain and an N-terminal tail which extend outside of nucleosome and present basic characteristic due to numerous lysine and arginine residues. So when we see the diagram below or the image below, here we can see there is this histone protein and there is protruding N-terminal tail which will have numerous lysine and arginine residues where the histone modification occur. That is the patterns, the acetylation, methylation, sumolization will occur in this N-terminal tail. So this is how the arrangement of the octamer is, histone octamer, around which the DNA is coiled, and this darker green tails are the histone tails, which protrude out from the nucleosome. So what are the types of modifications that can take place on histone tail? That is, addition of the acetyl group will result in acetylation. Likewise, there will be methylation, phosphorylation, ubiquitination, and sumolization. Again, here there is an example where we see that there is an histone octamer and the poly N tail, where there are these residues, arginine lysine residues, where there will be addition of these groups, that is methyl, acetyl, likewise. So here in the below area, we see that There are some enzymes, that is histone acetyl transferases and histone deacetylases. So when there is histone acetyl transferases, it is going to relax, or it will open the chromatin. It is going to open the closed chromatin. The reverse is also possible. That is, the open chromatin can be closed with the help of histone deacetylases. So it's not always that this functional groups, that is addition of the functional group, is going to result in the opening of chromatin or closing of chromatin. It totally depends upon the functional group and the charge of the functional group, because even the histone proteins are charged, even the DNA molecules are charged. So addition of this functional groups can result in variability in the charges, and likely it can result in a closed chromatin or to an open chromatin. So when we study the methylation patterns, it is seen that the methylation results in the neutral charge, and there is increase or close packing or close chromatin is formed. When there is acetylation and phosphorylation, we see that there are negative charges, so therefore there will be repulsion of the histone protein and the DNA strand. Therefore, there will be unwinding or opening of the chromatin. So this is what I have explained in the previous slide as well about the histone acetyl transferases and histone deacetylases. Now, methylation patterns can be monomethylation, dimethylation, and trimethylation. So in the literature, when you see H3K27Me3, it has some meaning. So what does this mean? Now, H means histone, K means the lysine residue, Me means the methylation. So when I want to read this, it will be the trimethylation at the 27th lysine residue of histone 3 protein. I repeat, it will be, we will read this as trimethylation at 27th lysine residue of the histone 3 protein. Now, here are some examples. Again, H3K4Me2 is upregulated with increasing stages of oral squamous cell carcinoma. H3K9AC is upregulated in mucoepidermoid carcinoma, whereas H3K9Me3 is upregulated at tumor invasive front and is correlated to aggressive characteristic of mucoepidermoid carcinoma. Likewise, when we want to study the downregulations, like H3K4Me3 is studied downregulated in oral squamous cell carcinoma. So these were the few examples for histone modifications. Now we need to, the last modification that is there is microRNAs. So microRNAs or mature microRNAs are single-stranded RNAs, which is comprises of 22 nucleotides. They are under RNA interference system, which is microRNA and small interfering RNA. So microRNAs are abbreviated as miRNA. So it regulates negatively the expression of different target genes post-transcriptionally. MicroRNAs function via base pairing with complementary sequences within mRNA molecules. That is, it is going to affect the messenger RNA. MicroRNAs are going to hinder with messenger RNA. And as we know that the messenger RNA is an important component of the central dogma. So therefore, there is going to be alterations in the gene expression if these microRNAs are affecting the messenger RNA. So let us study how microRNA biogenesis is carried out. That is, microRNA gene has been transcribed to primary microRNA, which is read as pre-microRNA, that is pri-microRNA, which is then cleaved to precursor microRNA, which is read as pre-PRE microRNA. Again, it is cleaved to microRNA duplex. Then there is unwinding and there is assembly, risk assembly. Now, what is this risk? Risk is nothing but it is abbreviated. It is RNA-induced silencing complex. And after this unwinding, there is formation of a mature microRNA. And this mature microRNA is going to affect your messenger RNA by translational repression, that is silencing, or mRNA cleavage, that is degradation of the messenger RNA. So this is how microRNA biogenesis and effect on the messenger RNA takes place. So there is again a nomenclature for microRNAs. So when you see such example, that is HSA MIR 26A2. So each has some certain meaning. That is HSA because the species is Homo sapiens, that is humans. Had it been Drosophila, then we would have taken it as D, or virus, then we could have taken V. Now, small r represent precursor microRNA and capital R represent mature microRNA. 26 is the number or the order of the discovery. That is, if it has a higher number, means it has been discovered latest, and if there is a lower number, then it has been a very older discovery. Small a is the variation and 2 is the identical microRNAs. That is, it represents the number where there is identical microRNA found in different locations of genome. It is a large number would mean a cluster. Means the larger the number, it means it is the similar microRNAs are present in a cluster. So there are certain microRNA databases available online, which we can use for interpretation of the data. So this is the library, microRNA library in the literature. So there are few examples also, like how microRNAs are studied in carcinogenesis and neoplasias. Like microRNA 21 is upregulated. Now, one must remember that microRNA, which is initiating a cancer or associated with carcinogenesis, is known as oncomere. So microRNA 21 upregulated in head and neck squamous cell carcinoma, whereas 31 is studied in, again, oral squamous cell carcinoma. MicroRNA 489 is upregulated in amyloblastoma, and there is panel of other microRNAs which is upregulated, like microRNA 135b, then 31, 592, and 944, which is again upregulated in amyloblastoma. Now there are some downregulated microRNAs as well, like microRNA 375, which acts as tumor suppressor in oral squamous cell carcinoma, and again the panel like 338. 3P 23A to 03 is studied in osteosarcoma. So there are methods to study epigenetic modifications. Now this is the paradigm. This is the general process that we need to understand can be harbored for diagnostic procedures like genomics, transcriptomics, proteomics, and metabolomics. So to study DNA methylation and histone modifications, there is gene-specific profiling and genome-wide profiling. In gene-specific profiling, there can be immunohistochemistry, bisulfite PCR, or methylation-specific PCR, or pyrosequencing. When you want to study genome-wide profiling, it is microarrays, next-generation sequencing, mass spectrometry, and chromatin immunoprecipitation. MicroRNA detection methods are immunohistochemistry, gel electrophoresis, RNA sequencing, quantitative reverse transcriptase polymerase chain reaction, and microRNA microarrays. So to conclude, understanding epigenetic modifications is important. Most tumors are a true outcome of epigenetic modifications. The reversible character of epigenetic modification is currently explored in preclinical research for therapeutic purposes in different types of malignancies, including oral cancer. These are my references. So there is a small exercise. I will give you 15 seconds. You just assess yourself and correctly identify the option. So here we see that you need to identify the correct CPG motif depicted. Yes, the option will be D, because the motif is always read from 5' to 3' end. So the A and B options are ultimately crossed out. The C and D remains, but in C we see that there is no C and G in the sequence. So C and G sequence is present in the D option. So the answer is D. The next is correct statement for given illustration is, so H2K2Me3. So you need to identify the correct option. I will give you 10 seconds. Yes, the option will be B, because it is histone 2 is been trimethylated at second lysine position. I hope this presentation was helpful. If they ask you anything and you don't know, just say it's due to epigenetics. So I would like to thank Dr. Gargi Goswami ma'am and Dr. Geet Priya ma'am for giving me this opportunity. I would also like to thank organizing committee and my patient listeners. Thank you, everyone.